So first and foremost, as any lab, depends on a very good workforce. So we've got an excellent team, a Celtic team for the most part, Hugh, but very interested in their rugby and the brain damage associated with that, but a good team. And for those of you that are students in the audience, I think one of the things that we've got to focus on is that research is about ideas. Everything else is grit and grind. And those of you that are doing masters, to those of you that are engaging in PhDs, I know that life sometimes feels a little bit like this. And if you're lucky enough to be offered a postdoc, life changes again. And if you really focus on your science and you communicate well and you publish your work, relentlessly working on that H-index, one day you could become Superwoman, or you could become Superman, Mike. And as David was saying earlier on, Journal of Physiology, it's a fabulous go-to journal. I think it's one of my favorite journals. For those of you that are interested in the topic I'm going to talk about today, here's a recent review. For those of you that are interested in a far more entertaining read, well, Nick Lane has done a marvelous service, I think, to this area. Very, very interesting. And science, for those of you that aren't entirely convinced, is actually great fun. And I think that's one thing that the applied and the extreme side actually brings out, is that you can combine adventure with good science. So, back to this individual here. So the take-homes really is that oxygen has driven biological evolution. I'm going to focus unabashedly on oxygen as a molecule. Humans are obligate neurobes. We've evolved to be entirely dependent on oxygen. But yet oxygen, it's a quantum paradox. It's a toxic, mutagenic, deadly gas. Fascinating gas. The brain definitely generates free radicals, and we're going to focus on the good, the bad, and the ugly, but as a half-pint full Celt, I'd rather focus on the hormetic basis, the good. And science is fun. Okay, so part one, setting the scene. Evolution of the obligate neurobe. Well, if you go back in time, quite some time, in fact, about 4 billion years, and you look at the changing paleoatmospheric oxygen concentration, you note that around about the time of cephalization and during the Cambrian explosion, you've got these oxygen pulses. And if we blow up the Cambrian explosion from about 600 million years ago, the fascinating feature about this is that every time there's an oxygen pulse, it's coincided with an explosion of life. And in fact, the cardiologists can thank this, and the neurologists can thank this. A tripling of brain volume in a very short period of time, in less than 2 million years. The brain, the human brain, has become very, very large. That's a good thing to some extent. But enjoy it while it lasts, because in around about 3,600 years, the oxygen concentrations are slowly depleting. And living at La Rinconada, which is the highest habitable place, and it's a very tough place to live in Peru, at 5,340 meters, our ambient fractional concentration of oxygen will be equivalent to that in approximately 3,600 years. But note, there'll be no oxygen in 4,400 years, based on recent estimates from Scripps. So enjoy it while it lasts. Big focus on CO2, but let's spare a thought for oxygen for a second. That's not necessarily a good thing. The tripling of brain volume means that for the Americans in the audience, our brains are like a Hummer. Not like a European Cinquecento, but like a Hummer. It's a gas-guzzling energy hog. Coming in at 2% of our body mass, it consumes a disproportionate 20 to 25% even of our basal energy budget. And we know the reason for that is when we get deep inside the mitochondria, we've got extraordinarily high rates of neuronal ATP transmission. In fact, so high, 4.7 billion molecules of ATP are being turned over and hydrolyzed every second in your brains right now. Huge energy budget. This is the first image of a 3D CT MR scan looking at glucose metabolism. Look at the brain. Glucose uptake, exclusive glucose uptake in the brain. It's very reliant on this fuel in order to fuel synaptic transmission. So the brain is very, very oxygen hungry. But it's incredibly vulnerable to failure, and it's something that's really bothered me from an evolutionary perspective. If you assume an average cerebral capillary PO2 of 25 torr, a content of 30 nanomolar per milliliter. The brain at rest is consuming 30 nanomolar per milliliter per second. You can see that the aerobic energy reserve is going to last us a second. And we know when we deprive the brain of oxygen for a prolonged period of time and we initiate these irreversible death cascades, that life can actually become very, very difficult. And in fact, this is an experiment that was tested as part of the Red Wing studies. Without question, one of the most unethical studies I've ever read. And you've only got to look at the title. Well, you can look at the model as well, of course. Acute arrest of cerebral circulation in man, 1943. 3 clinicians published this work, and they asked a very simple question. If we applied bilateral carotid arterial occlusion and cut off all blood supply to the brain, what happens to schizophrenic patients, in this instance, over prolonged periods of time, up to 200 seconds. It's fascinating. For all of the wrong reasons, of course. And we know that this individual, on average, if we were to perform this, so 600 millimeters of mercury within 0.2 of a second, complete carotid arterial occlusion, 6 to 10 seconds before the individual then becomes unconscious. So how have we evolved with a brain that's so oxygen hungry, yet is so exquisitely sensitive to failure? It's always bothered me. There seems to be some sort of a paradox there. Of course, we don't do those types of experiments. We look at some of the high altitude models, and I've always been fascinated by the heterogeneity of the response to high altitude. So this is Parinacota in Chile. It's just about there at base camp, and this highlights or illustrates, if you like, the heterogeneity of the response. We've got Chris Imray standing there, rather old, joking, well seasoned. We've got Emily here. She's never been to altitude before, and we've got an incredibly expensive postdoc that cost me a fortune to get up the mountain to help me do all the work, and there he is, completely and absolutely incapacitated with severe altitude sickness. So Kevin has got early high altitude pulmonary edema. All the gear, no idea, in simple terms. So why do we respond so differently to this environment? And my focus really is on the brain, how the brain senses and tastes oxygen. But before we get there, just a brief word from the expert. This is my son, Luca. He's not the last universal common ancestor. It's just that my wife likes the Italian name. And it's publish or perish in my house. So we've got a paper in preparation at the moment for Nature. So just very simply, any atom or molecule that lacks one or more electrons in its antibonding orbitals is known as a free radical. And here's your free radical lacking that electron. Here's a sacrificial antioxidant, and it looks to stabilize the species, the atom or the molecule. And this has had a bad press. This can cause membrane destabilization and cause damage. And they come in all shapes and sizes. And when we look at free radicals, we look at and we calculate the 1 electron reduction potential. Some can be incredibly aggressive, incredibly excitable, like the hydroxyl radical, with a billionth of a second lifetime. This is this chap here. Or we can have, for example, the superoxide radical, which is incredibly domesticated. In fact, the last thing superoxide is super. It's a very, very unreactive species, but it's involved in cell signaling, and that's what we're keen to look at here in vivo. When you get deep down into the oxygen molecule, you can see that it's lacking 2 electrons in its antibonding orbitals. So this is a diradical. Oxygen is a free radical. It's a diradical, as you can see there. And too much oxygen, we know, pure oxygen can cause irreversible pulmonary fibrosis. It's toxic. And the interesting thing is, based on the Pauli exclusion principle, thermodynamically, and it's an evolutionary quirk if you like, oxygen cannot, because of this exclusion principle, accept these 2 electrons simultaneously because they've got parallel spin. It accepts them one at a time, very sluggishly, and it's good news for you and I, because if it wasn't for that thermodynamic limitation, we would all combust spontaneously in 20.93% oxygen. You take the most basic bacteria, they swim away from a high PO2 in a Petri dish to a low PO2. They don't like a high PO2. And perhaps the pressure gradient in terms of the oxygen cascade from into the mitochondria is not just to drive a PO2 gradient, but it's to protect some of the more sensitive organelles from the full flux of oxygen. We know the higher we live, the longer we live. Endosymbiosis is a great example. Lynn Margulis's work demonstrating that there's a symbiotic relationship here between blue-purple bacteria and the mitochondria to constrain and limit free radical formation. And we know that the best diving species, the most hypoxia tolerant of all species, switch their brains down in terms of oxygen consumption when they're under pressure. We humans, unless we're free divers, we do the exact opposite. So there's a bit of the bad and the ugly there. So the second part, we set the scene. Now let's set the sense. How do we as humans taste oxygen? Well, like every physiologist, you've got to have good ideas, techniques, and models, and I always like to think it's important to be ambitious. Let's get to the organ of interest. Now, evolution has favored coupled oxygen transport, and if we go over time, as time has gone by, the molecular machinery to taste or sense oxygen has become increasingly complex, from simple nematodes all the way through to mammals. And we know that as oxygen concentrations decrease, typically in normoxia, prolyl hydroxylase activation of HIF commits it to proteasomal degradation. And in hypoxia, PhD inactivation and the accumulation of HIF and the binding to the hypoxic response element drives polycythemia. We see mitochondrial biogenesis and capillarization. And all of these are designed as very sophisticated systems to try to defend falling oxygen concentrations. The red blood cell is floating around the circulation, sensing, tasting hypoxia, and a variety of bioactive nitric oxide metabolites, be it ATP, S-nitrosahemoglobin, or nitrite, are released into the smooth muscle to affect vasodilation. And the fundamental stimulus is that oxy to deoxyhemoglobin allosteric transition. Very, very interesting. But if I wanted to sense oxygen, I'd want to be a Ferrari. I'd want to have something as a very short half-life as that upstream molecular signal. And thermodynamically, you're going to find none better than a free radical. Some of these species have got half-lives as low as 10 to the minus 12 of a second. So they've got incredibly short half-lives, which makes our job very difficult trying to capture them. So we rely on this technique, EPR spectroscopy. It's the only technique that allows us to measure, characterize, and detect the radicals. And we use a variety of different spin traps that we can add to blood, we can add to tissue, or any media. Some of them we can tag to mitochondrial specific free radical formation. We use ozone-based chemiluminescence as our gold standard to measure the bioactive nitric oxide metabolites, both in plasma and in the erythrocyte. These are the types of signals that we detect in the nanomolar range. And we focus on measuring nitrite, S-nitrosahemoglobin, because the field has been polarized to some extent in terms of what camp you actually ascribe to. But the bottom line is that the erythrocyte is armed with a whole range of tools to be able to sense and taste the oxygen. Here's a typical model. We measure flow across the brain using duplex ultrasound. We can catheterize internal jugular vein for cerebral venous effluent and the arterial circulation for inflow. Here we've got ephemeral arterial line, ephemeral venous line, thermodilution measuring flow across the leg, a jugular venous and a brachial art and measuring flow across the brain. So we can actually capture our site-specific samples, subject it to the measurements. These are just some of the measurements that we typically make in terms of free radicals and bioactive nitric oxide metabolites. We can exercise our volunteers, we can make them profoundly hypoxic, and then we can do our Fick exchange calculations. So we can determine mass specifically how much, what concentration per 100 gram tissue of brain is the brain generating or consuming for that matter. This is a typical example from Ben Stacy Spielberg. He spends a lot of his time doing movies as opposed to writing papers. Sorry, I'm only joking, Ben. But here you can see a typical experiment. We've got the vertebral arteries being measured here, the posterior circulation, the ICA, the anterior circulation. Tony here is collecting bloods, AV across the brain. We've got blood gases that are being run here. We've got a whole range of other measurements that are going on. And we're dynamic end tidal forcing So we're maintaining carbon dioxide, we're pulling that out of the equation and focusing specifically just on the role of oxygen here. So a typical experiment, but we can go high as well. This is the wonderful thing about this area of research. We can combine the science with great adventure. And for perhaps the younger members of you, this is a wonderful way to write grants and actually do a bit of traveling and experience something a little bit different. So we've been across to a whole range of different places and done some really interesting experiments. But of course, it's got to be underpinned by good laboratory-based science. We've worked with some really crazy models, some very interesting models with the high G models, and being able to sense very quickly cerebral hypoxia, of course, because you get yourself into trouble very, very quickly. Some of these fighter pilots as part of their training experience. We apply this. We heard a little bit about Hugh talking about some of the clinical applications, the biomedical applications. We apply this in some of the surgical settings with low flow, high flow, and the changes in PO2 there. We see an awful lot of this in Wales, probably as a result of fish and chips. This is a carotid endarterectomy being shelled out here by the surgeon. And we are also interested in some of the superhuman models. So with a single breath hold, this chap can last for 11 minutes, 54 seconds. So just one breath hold. And if you do the kinetic calculations in terms of oxygen depletion, he shouldn't be able to do this for longer than just over 8 minutes. But yet he goes for almost another 4 minutes. If you put him on 100% oxygen for 10 minutes and then 3, 2, 1 breath hold, you can double that to 24 minutes. So phenomenal. And we've been able to instrument and go across brain in collaboration with Phil Ainsley at University of British Columbia. This is something I don't suffer with. Giraffes, really absolutely fascinating. And we've been working recently with giraffes, with Nils Secker in Copenhagen. This is a giraffe that's being instrumented. We can measure flow across the brain. We can sample across the brain. We can measure using NIRS, cortical oxygenation. And when the giraffe stands up really quickly, if you watch giraffes drink, they stand up very quickly. And there's some degree of regional hypoxia, interestingly, and we're interested in mapping this out. So what we're doing is, actually, this is a giraffe under a general anesthetic that's instrumented and a fashioned Heath Robinson, if you like, pulley system to mimic what they do when they're drinking. So as a giraffe bends down, very ungainly, and then stands up very quickly, how it does this physiologically is really quite fascinating, and some degree of cerebral hypoxemia we feel exists during this maneuver. And beyond super, of course. Coming in at the silver medal, the sea turtle, bronze, the Canadian bar-headed goose, and tasty and tough, 100,000 minutes of pure anoxia. The common crucian carp is, for me, the #1 performer. Never once had a stroke in his life. But what we do find is that the brain is remarkably robust. When we drop arterial oxygen content, it maintains flow in all of the models that we've exposed the brain to in terms of severe hypoxia, such that the cerebral delivery of oxygen is well preserved. So we can't trick the brain into under-delivering in terms of oxygen. It's ruthlessly well defended. We've done this again in collaboration with Chris Willey and Phil Ainsley at UBC. We can get these freedivers to give us an 11.5-minute breath hold. We can go across brain. We can demonstrate at the end of an 11.5-minute breath hold that there is no AVO2 difference across the brain. So the only way they can get oxygen into the brain is by flowing, and they flow jolly well, such that they defend the delivery of oxygen. And interestingly, defense seems to be specific through the vertebrates, through the posterior part of the circulation, which phylogenetically speaking is the older part of the brain, far older than the anterior part of the circulation. So there seems to be region-specific defense. We know from the AV studies that when we go across the brain, that the brain generates radicals in hypoxia. This is the arterial systemic circulation. And then when we look at the venous outflow across the brain, we see about a 5-fold greater release when the brain becomes hypoxic. So we feel confident using the best technology, being as direct as we specifically can, that the brain in hypoxia is generating more radicals. And as I said to Hugh, this took about 7 years to get published originally years ago because people just didn't get it. How on earth can less oxygen generate more oxygen-centered radicals? Now, of course, we know that hypoxia is a common stimulus generating radicals. In separate studies, we've taken our volunteers, we've put them into MRS, magnetic resonance spectroscopy. We've measured intracellular PO2, cytosolic PO2 as a surrogate measure of mitochondrial PO2. We've demonstrated that we have a reduction in skeletal muscle PO2, the mitochondrial cytosolic PO2, and we've generated a greater concentration of what we feel is a UB semiquinone radical coming from the mitochondria. So we think perhaps unifying this is that the brain, the cerebral mitochondria, are generating these radicals that we can then pick up in the circulation as it leaves the brain. CMRO2 seems to be well defended in severe hypoxia. When we calculate mathematically our mitochondrial PO2, of course there's a reduction, and we've actually mapped the output of radicals across the brain in direct proportion to the reduction in the calculated mitochondrial PO2. So as the PO2 is starting to drop, radicals are being spat out of the brain. Is that good? Is it bad? Or is it ugly? Well, the cellular studies tell us that mitochondrial free radical formation, in particular from complex 3 in terms of superoxide, though there is still a great deal of debate about this, inactivates the PHDs and drives this HIF pathway. And again, they're signaling molecules that are upstream, and they're acting very, very quickly to switch on this oxygen salvage pathway. And we're currently looking in the erythrocyte as well at the anion exchanger, because we feel that that's subject to redox regulation as well. So radicals are everywhere. We're just trying to dissect out their potential contributory role in controlling and sensing oxygen here. Experiments that we did quite some time ago now, where we knock radicals down at rest using an antioxidant intervention, and we ablate the generation of exercise-induced radicals. When I originally did this experiment, I thought that we were going to get rid of these nasty beasties by prophylactically knocking them down. But we demonstrated, in fact, that blood flow was halved when we got rid of the radicals. So that then, I started to think, well, is there a hormetic basis to the generation of some of these oxygen-carbon-centered radicals? And we've since done studies in older individuals where we've infused intraarterially high dose, millimolar dose of vitamin C. We make them hypoxic, and we've identified that we see a suppression with an antioxidant on board of these hypoxia-induced genes. So again, the radicals are there potentially driving some of these downstream responses. And a little bit like the Large Hadron Collider, where you've got particles that have got a half-life of 10 to the 67 of a second, billions and billions of pounds, I really feel that if we can look at some of these radicals and develop the technology more so that we can really look at super short half-lives and detect these femtochemical species, we'll understand this process a little bit more clearly. They're not just simply bad or plain ugly. They're there as sensors. And a new area of physiology with a father who's a particle physicist, of course. Sports science, medicine, they never come into the equation. Mathematics is the only fundamental science. And so for a bit of fun, just to please him, I guess, more than anything else, physiology, I think we're in a domain now where we can apply mathematics to some of these very, very short half-life species. And these radicals potentially could be acting as quantum triggers. They can be anywhere in the cell at any point in time in terms of superposition and entanglement. So they could be switching things on without any boundaries. And so knocking them down might not necessarily be a good thing. They're there for a reason. They were present in Luca, not my son, the last universal common ancestor. And like everything else in life, I guess it's a matter of balance. And just finally, just a word in terms of who the greatest gas is. Well, I focused almost unicentrically on oxygen. We've heard a little bit about carbon dioxide. The allosteric shift from oxy to deoxy, I think, is a fundamental signal. It's a fundamental stimulus. Nitric oxide is definitely up there, no question about that. CO2 is unequivocally up there. And we've just done some recent studies where we've been able to clamp CO2 during hypoxia, so people don't become hypoxically hypocapnic, they become hypoxically normocapnic. So we pull CO2 out of the mix. And what we've identified is that CO2 is really affecting nitrosylation. and it's a real complication, such that part of me is now thinking whether or not this is driven by CO2 and far less by oxygen. But that's, I guess, a separate presentation for a separate day. One final thing. I snuck this slide in because I was having a chat with Hugh earlier on. I've talked to you about this almost paradoxical lack of oxygen reserve for the neural governor. How on earth have we evolved with such an oxygen-hungry brain, with such desperately meager oxygen and energy reserves? Well, I don't know if you've seen this study. We're currently working with Nenad Seston over at Yale. Domesticated pigs that are destined for food, so FDA approved, decapitated for food. The brains are removed. 4 hours after the brains were removed, they were surgically positioned on a transcorporeal ex vivo reperfusion system. And 6 hours after being set on the reperfusion system, we're not entirely clear what the perfusate is, but function could be recovered. So 4 hours after the pigs had been decapitated, 6 hours post setting up on the cerebral perfusion system, such that you can see oxygen consumption, there was glucose extraction, lactate formation, and pharmacological vasodilation. So there's a restoration, if you like, of cerebral function way beyond what we would traditionally consider as death. Very interesting. So that's given us a clue that the brain holds this hitherto underappreciated, totally unexpected, and thus far under-examined oxygen reserve. So there's something there that we're not understanding. Hardly any neuroglobin, hardly any oxygen binding proteins, but there's something there with the capacity to hold on to oxygen, this precious molecule. And this is questioned, the medical interpretation now of brain death. And it's very, very contentious, very, very exciting. So, too many ideas to think about, but this is the fun of science. Feels a little bit like that most of the time, I must admit, Mike. But hopefully it's given you some ideas to explore. We've got a few postdoc positions that'll be opening up in the lab soon. Never a lone journey. Lots of good colleagues. A special thank you to Phil and the team at the University of British Columbia that will be talking later on today. And thank you for your attention.